So, hello everyone, a very good evening to all of you. I hope so. I am audible to all of you, and the screen is also visible to everyone. I welcome you all to another session of Brahmastra Series, and today we are discussing about Unit 4. That is taken into account two important topics from Unit 4. In fact, when I say Unit 4, overall, all the four subunits are very important, whether you consider signaling, cellular communication, cancer, and immunology. But right now, we are taking only two topics, that is cell signaling and cellular communication. So let's start with the session now. The first question to all of you over here is, which one of the following statements about the nuclear receptor superfamily is not true? The receptors are always cytosolic, where they remain associated with heat shock proteins and have variable ligand binding domains in the N-terminal region. Receptors have characteristic repeat of C4 zinc finger motif. Third, the receptors are either homodimeric or heterodimeric, and in the absence of their hormones, the heterodimeric receptors repress the transcription when bound to their response element. And the fourth one is, receptors have a unique N-terminal region of variable length that contains the NLS, nuclear localization signal, between the DNA and ligand binding domains. So look into the question carefully and get into an answer. And I want you all to respond to questions that is being displayed on your screen. Yes. So basically, when we speak of cell signaling, we have got around 4 to 5 signal transduction pathways, right? And I have just picked up something that is important from examination point of view. So nuclear receptor family, once upon a time, you used to get good number of questions from this topic in the form of 4 marks. But yeah, recent past, they have shifted to GPCR, RTK, JAK-STAT pathway like that. But still, let's have a look onto the nuclear receptor superfamily. Yes, can I get some answers for this question? What should be the answer according to you? And remember, here they are asking you which one is not true. So never get confused between whether they are asking true or not true. So can I get some answers from everyone? Yes? Who all are attending right now? Can I get some answers from your side? And this is like a very simple signal transduction pathway. Without much complications, unlike your GPCR, where it involves a lot of proteins, lot of enzymes, downstream proteins are involved to carry out the signaling process. So here you have got in fact, I must say, not much proteins involved over here. So what should be the answer for this question? So let me give you the answer. I hope so. You will be able to connect it when you see the answer. The correct answer over here is statement 1. Now, why the statement 1 is incorrect? The receptors are always cytosolic by nature and they remain associated with HSP. They have variable ligand binding domains present in the N-terminal region. So why this statement is incorrect? Because of this one terminology, always cytosolic. So what should be the rectified answer of this question? See, when I say a typical steroid hormone receptor, This is your red color one, indicates the hormone receptor, which will be having the ligand binding domain, the DNA binding domain, and the connecting link between them is the hinge region. Okay. So here if you see, this is the model of a steroid hormone receptor. So receptor means what? It is a kind of protein. So it will be having N-terminal region and it will be having C-terminal region. So towards the N-terminus, you'll find the domain 1, which is known as a variable domain that is responsible for its interaction with other transcription factors. Domain 2, which is known as the DNA binding domain, and it is enriched with zinc finger. Now, what is a zinc finger? It's a kind of motif. Then we have domain 4, where our ligand goes and binds. So we can call it as a ligand binding domain. And the domain 4 is where the dimerization takes place. See, whenever a ligand goes and binds to the corresponding receptor, obviously there undergoes a dimerization process. So that happens on the domain 3. Zinc finger is a motif. Motif in the sense that helps in the interaction with the protein. So if you see, this is your zinc finger structure. So zinc finger structure is basically coordinated with 2 zinc ions and 2 cysteine residues. So through this, they will be interacting with the DNA. Okay. Now imagine this receptor sitting on the DNA. So as you can see in this image, this is our DNA. The red color one indicates your receptor. So receptor is having the ligand binding domain, DNA binding domain, the connecting link, the hinge region. So how they will be connected to the DNA? With the help of zinc finger motif. So since in a single monomeric unit, you will have 2 zinc finger sites. So when I say dimer, they will be having all total 4 zinc finger motifs will be there. So they will be interacting with the hormone response elements which are present on the DNA. Okay. So we can classify the intracellular receptors into 2 types. First one is type 1, present in the cytoplasm. Usually they are bound with HSP, that is your heat shock proteins. So all the sex steroids, glucocorticoids belongs to this category and they form homodimers. Second one is type 2, which is present in the nucleus, often found associated with the DNA. So vitamin derivatives will be serving as a ligand over here, like thyroid hormones, vitamin D, vitamin A, like that. And in this case, they can form both homo and heterodimers. Okay. So if you look into the pathway over here, so the type 1, as I told you, they will be bound with the HSP protein. When you don't have a ligand bound to that, when you don't have an hormone bound to that. Now, when this hormone goes and binds to the receptor, HSP will be released. Now, the ligand receptor, they will undergo a dimerization process. So, because of this dimerization, NLS will be exposed. So, through the nuclear pore, they will enter inside the nucleus, binds to the hormone response elements present on the DNA. So, you will be finding, since they need to undergo gene expression, so obviously the polymerases and the coactivators, everything will come together, right? Then only the mRNA synthesis takes place and finally the protein synthesis will be taken place. So that is the case of type 1. But what about type 2? In this case, the receptors are already sitting on the DNA. So there is no requirement of translocation. So the hormone directly goes and binds to the receptor. And the way here you had the HSP present, here you have the co-repressors present. So when your goes and binds to the receptor, the co-repressors will be released. The transcriptional machinery, that is the polymerases and the coactivators, will be recruited over here and mRNA synthesis takes place and finally they will go outside. This results in the protein synthesis. Okay, so that is how your type 1 and type 2 works. I hope so I'm clear to all of you. Can I get some response? Yes, am I clear to all of you? Nuclear receptors is clear to all of you? Type 1 and type 2? Okay, good. So next we have this image. So with reference to the signaling pathway shown here, you have to identify what does A, B, C and D codes for. So can anyone tell me what is this pathway indicating? What does this pathway indicates? What does this pathway indicates? Which signal transduction pathway is shown in the image over here? Where a ligand goes and binds to the receptor, some phosphorylation takes place, A, B, C, D getting activated, and this results in cell proliferation. Good evening, Sumita. So what will be the which signal transduction pathway is it indicated over here according to your understanding? Yes, Sumita, can I have an answer from your side? How many signaling pathways do we know? We have ligand gated ion channel, we have receptor enzymes, receptor without enzymatic activity, then we have the Jackstat pathway, then we have the GPCR. Absolutely correct, Sumita. So this represents your tyrosine kinase pathway. So can you identify what does this A, B, C, D indicates over here? Can you identify what does this A, B, C and D indicates over here? So requesting others to please participate and give the answers. As I always tell you, yesterday also I had mentioned that this is for your benefit. This is for boosting your confidence within you. So try to give the answers, everyone. Okay, I got an answer from Sumita. Can I expect any other answer? Can I expect some more answers from your side? So this is your RTK pathway, which is also known as receptor tyrosine kinase pathway. The correct answer over here is 1. Now, how 1 is the correct answer? So this indicates your GRB2. GRB2 will be activating SOS. SOS will be activating RAS GTP. And then RAS GTP in turn will be helping in the activation of a transcription factor known as ERK. So that is how it goes. So after GRB2, RAS GDP will come, but RAS GDP is the inactive version. Okay, it has to be converted into RAS GTP form. Then only you'll get the correct answer over here. And also MEC, ERK, both are transcription factors. So in case instead of this, if GTP is written, yes, we can consider this second option. But since it is mentioned as RAS GDP, you have to think of first option. So see, in this case, you can see we have got the insulin, which goes and binds to the corresponding receptor, that is an heterodimer, alpha and beta. So it is in your beta subunit where the tyrosine kinase present, alpha subunit where the insulin goes and binds. So upon binding, autophosphorylation takes place. This activates or recruits the substrate, that is IRS1, to the position. So they will be interacting with each other through their specific domains. If you are aware about specific domains, then yes, it is your PTB domain. which is responsible for interacting through phosphorylated tyrosine residues. Okay. Now this phosphorylated IRS1 interacting with GRB2. GRB2 in turn will be interacting with IRS1 on one side and SOS on the other. As mentioned yesterday, SOS is a kind of a JEF. JEF means some exchange factor. So what is the exchange going on here? As you can see, GDP is going out, GTP is coming in. So SOS catalyzes this exchange. Now you got the RAS GTP. Upon activation, RAF protein will be activated. Now this RAF protein in turn will be phosphorylating the first transcription factor MEC1 on two serine residues. Okay. And upon the MEC phosphorylation, they will be phosphorylating ERK on a threonine and tyrosine residue. So why they are phosphorylating? Because phosphorylation leads to their activation. Now, once your ERK is phosphorylated, they will be released into the nucleus, combines with the transcription factors, and they will carry out the necessary protein synthesis. So what is the necessary protein synthesis, or what is the function of insulin? Since it is a molecular level, it is related to cell division. Okay. So that is how the approach of the question. So you can see exactly the images was asked in the question. Moving on to our third question. Now this is related to your GPCR pathway. So the signal transduction pathway involved in glycogen metabolism triggered in the liver by the hormone epinephrine involves the following steps: activation of G protein by the receptor, protein kinase A activation, second messenger generation, adenylyl cyclase activation. Identify the correct sequence of the signaling pathway. Identify. Very good evening, beautiful colors. So today you joined late. Can have a look into the question and get me an answer for this question. Yeah, no problem. Just have a look into the question. This is related to your GPCR pathway. How can I say that this is a GPCR pathway? First thing is that they have mentioned as epinephrine as their ligand. And here G protein is mentioned. So obviously we can predict that this is your GPCR pathway. Okay, Mahesh. What about the others? Okay, Sumita. Beautiful colors. What is your answer then? Okay, Sumita. So you have changed the option. Okay. So let's check it out. What is the correct answer over here? The correct answer over here is, first thing is that your G protein is getting activated. So once your G protein is getting activated, AC will be activated. AC in turn will be leading to the activation of second messenger. And second messenger in turn leads to the activation of protein kinase. So what should be the answer over here? It should be 1, ADCB. Always remember one thing. First, when a ligand goes and binds to the receptor, right? The first protein to be activated will be the effector protein. So what is the effector protein over here? Adenylate cyclase. Now what is the function of adenylate cyclase? To catalyze the process, because it is an enzyme. So enzyme function is to catalyze the process. So in that case, it helps in the conversion of ATP to CAMP. Now after CAMP, the next protein to be activated will be the protein kinases. So once your protein kinases are activated, they will be going and phosphorylating the target protein so that they will generate a response. That's the reason ADCB is the correct sequence over here. I hope so you understood why option 1 is correct, everyone. Right? So this is a simple GPCR pathway. The sequence, okay? Remember the order in this way. Yeah. So if you see this image, this is the resting phase. Resting phase in the sense we have got an inactive receptor. GPCR is present in the GDP bound form, accommodated with 3 subunits, alpha, beta and gamma, and obviously effector protein is also inactive. But when an hormone goes and binds to the receptor, see the difference in the structure of the receptor. There occurs a conformational change onto which your G protein can go and bind. So when your G protein goes and binds over here, your GDP will be released and GTP goes inside. Once the GTP goes inside, the subunits get separated. That is G alpha GTP on one side, beta gamma will be released on the other. So this G alpha GTP will go and bind to the effector protein. Now your effector protein is activated. So the signal transduction happens. Now if you want to terminate this process, simply the GTP can undergo GTP hydrolysis, hormones can be removed, and everything comes back to the previous setup. Okay. So this is how it goes. And remember, our G protein exists in 2 states. One is the GTP bound state, which is known as the on state, and another one is the GDP bound state, which is known as the off state. So why this is on, because it is having triphosphate over here. TP indicates triphosphate, so GDP with one gamma phosphate is present over here. So this gamma phosphate is stabilized by glycine and threonine on the 2 switch present on the G protein. So this becomes the active closed conformation. On the other hand, if the gamma phosphate is removed, as you can see a scissor sign over here, if the gamma phosphate is removed, the protein becomes open. So now what is left of? GDP. So this comes your off state or the inactive state. Remember this 2 forms. Then 2 enzymes that helps in the activation and inactivation. As I told you in yesterday's session also, Jeff is responsible for the exchange. So that makes your G protein GTP bound activated. On the other hand, we have GTP's activating protein that helps in the hydrolysis. So that will convert your G protein to GDP bound form, which is considered to be the inactive version of the G protein. And remember, G protein based questions are always expected in the exam. So never ever skip this part. Moving on to our next question. CGMP is produced from GTP by the enzyme guanylate cyclase, which exists in soluble as well as membrane bound form. So here are some statements related to signaling molecules that are associated with the signaling cascade. Atrial natriuretic factor causes natriuresis and diuresis by interacting with membrane bound form of GC. Guanylate cyclase, we can say GC. Nitroglycerin causes smooth muscle relaxation and vasodilation by interacting with soluble form of GC. Nitroprusside, they cause also same process, smooth muscle relaxation, vasodilation by interacting with membrane bound form of GC. And then we have again ANF causing natriuresis and diuresis by interacting with soluble form of GC. Identify the correct combination of statements among the following. So, you all might be aware about guanylate cyclase, which is existing in 2 forms, right? Membrane bound form and soluble form. So, based on that, this question is provided to you. So, one thing is for sure, if you have a keen observation, A and D both cannot be correct. One has to be correct, right? Because ANF can either bind to membrane bound or it can bind to the soluble form. Binding to both is not a possibility. So you can easily eliminate the option 4 from here. Now check out the other. So obviously one among them has to be correct. So either it should be AB, BC, CD also you can omit in that case. If you are sure that D is correct. Okay, so that will be based upon your deciding factor whether you are taking A or D taken into consideration. Yeah, check out and tell me. Is it a tough question? What should be the answer for this question? So I have given some clues also over here. If we can identify the correct combination from this given statements. Yes, Mahesh, beautiful colors, and Sumita. Can I get some answers? And others? I hope so. The previous question was clear to all of you. Okay, I got one answer, that is beautiful colors B and C. Anyone else would like to give the answer? No? Let's check out the correct answer over here. Actually, I have almost said you the answer. If you have listened to me carefully, I have almost said you the answer. The correct answer over here is 1 and B. I told you that one of them has to be correct. So either the ANF should bind to the soluble form or to the membrane bound form, right? So it is bound to the membrane bound form. Second is the nitroglycerin tablets. So these are involved in relaxation and vasodilation. Nitroprusside also have the same process, but instead of membrane bound, if it has mentioned soluble form, it would have been correct. So that's why statement 1, that is A and B, are correct. See, ANF is also known as atrial natriuretic peptide, which is a hormone released by the atria of the heart in response to stretching of the arterial walls. So, one of its physiological role is to regulate blood pressure and fluid balance. So, the mechanism by which ANF induces natriuresis, that is excretion of sodium in the urine, and diuresis, that is increased urine production, they are involved with membrane bound form of guanylate cyclase. So remember, they are related with membrane bound. Both this nitroglycerin and nitroprusside, they will act by promoting the production of CGMP. How? Through the activation of soluble form of guanylate cyclase. So that will be helping in contributing to their therapeutic effects related to cardiovascular conditions. So this is the image. Membrane spanning, and this is the soluble version. So membrane spanning, basically these are a homodimer single membrane spanning segment. So ANF can serve as a ligand, guanylin and endotoxin receptors also can serve as a ligand. So ANF, the receptors in the cells of the renal collecting duct, as well as it is also present in the vascular smooth muscles. So somewhere it is related to decreasing the blood volume, blood pressure as such. Guanylate is the peptide hormone produced in the intestine. So obviously their receptors are present in the intestinal cells. And they are also the target of bacterial endotoxin that triggers severe diarrhea as well. So that is the membrane spanning version. Soluble form, always remember the soluble form is bound with nitric oxide. And nitric oxide is produced with the help of one amino acid. What is the name of that amino acid? It is arginine. Okay. So it is a heme containing enzyme activated by nitric oxide found in smooth muscles of heart as well as blood vessels. Okay. So, nitroglycerin is a vasodilator used to treat conditions like angina pectoris and heart failure. That is the heartache. That is angina pectoris. And it is converted to nitric oxide in the body. So, nitric oxide can activate the soluble form of GC, which increases the CGMP production that helps in smooth muscle relaxation and dilation, resulting in better blood flow, thereby reducing the workload on the heart. Now, if you take up nitroprusside, they are also doing the same thing. They are converted into nitric oxide in the body and they will go and bind to the soluble form of GC. So, as usual, it increases the CGMP production, contributing to smooth muscle relaxation and vasodilation. So, what is the function of nitroprusside over here? To decrease the blood pressure. Okay. So, both of them are bound to the soluble forms with different functions. That is the difference. Okay. Next question. Some of the sequence of events involved in phototransduction in rod cells upon elimination are given below. So you have to identify the events in visual transduction during light perception. Hyperpolarization, activation of phosphodiesterase, decreased release of synaptic transmitter, activation of transducin, decreased cellular CGMP, and conformational change in rhodopsin. So here also they are asking you the sequence in which your GT pathway works. GT pathway is related to rhodopsin signaling, that is vision. Yes, Mahesh, you were correct. Now find out this answer. So GT pathway, again, when I say GPCR pathway, you have different types of GPCR pathway, right? So you have got GS pathway, GQ pathway, GI pathway, and then you have got the GT pathway. So if you ask me among this, what is the most important one? GT pathway. But again, it doesn't mean that if I say GT is important means from the rest of the GPCR, you won't get questions. You do get the questions, but many a times you'll find that questions do come from GT. But however, if we check the previous year question paper, you have also got questions from GS. In fact, the first question which I had asked regarding GPCR is GS. Then GQ pathway also you got. Compared to others, GI, you got less number of questions. So what should be the correct sequence of events of phototransduction event in rod cell? Any idea? Okay, Mahesh, I can see your answer. You have given 3 as the answer. What about others? What about others? Yes. Okay, Sumita. Beautiful colors. Do you have any answer to say? How about others? Anyone else would like to give any other answer? You have to be clear what happens during the light phase and what happens during the dark phase. Based on that, you have to get into a conclusion over here. So, whenever you get such questions, instead of going through this statements which is being provided to you in a rough page, try to mention what happens in the light phase and what happens in the dark phase. So, that will help you to get into an answer without undergoing any confusion. Okay, so all of you has given the answer. Let me check out what is the correct answer over here. The correct answer over here is 3. Absolutely correct. So let's check it out why 3 is the correct answer over here. So it started with FDBEAC. Conformational change happens in the rhodopsin, which activates the transducin protein. Then phosphodiesterase will be activated, after which obviously phosphodiesterase means it will break down the CGMP. So decreased intracellular CGMP, and then it leads to closing of the ion channels, that is your hyperpolarization, and finally decreased release of synaptic transmitters. Okay, how it is work? See, first thing is that we have got rhodopsin protein. So rhodopsin protein exists in two forms, 11-cis retinal and all trans retinal. So normally it exists in the 11-cis retinal form, which is connected to the lysine side chain of the opsin protein. So opsin is the protein part of the rhodopsin. So when the light hits the retina, what happens? There occurs a conformational change over here, which converts 11-cis retinal to all trans retinal moiety. So this results in the activation of rhodopsin protein. Right? Now apply this over here. In the dark, rod cells are active, CGMP is active, cation channels open, which results in generating action potential. This action potential helps in the release of positively charged ions, so we call it as a hypopolarization events. That releases the inhibitory neurotransmitter. Bipolar and ganglionic cells will be inactive, and this results in no vision. On the other hand, in the light phase where the rod cells are inactive, CGMP is converted to GMP. How? With the help of phosphodiesterase. Obviously, specific ion channels will be closed. No action potential, that results in hyperpolarization, and this causes the release of excitatory neurotransmitter. Bipolar and ganglionic cells will be activated, and this results in vision. So that is how it goes, the dark and the light phase. So whenever you get any kind of question like this, try to write it down. What is the difference between them? And after that, if you look into the options, you will be more clear with that. So this is the signaling pathway. So what happens here? This is the rhodopsin protein with all 11-cis retinal. So when light hits onto the rhodopsin, it gets converted to all trans retinal. And after that, the next protein to be activated will be the transducin. So transducin again belongs to GPCR family. So it is also having alpha, beta, gamma subunits. And there will be a GDP, GTP exchange. Now your transducin is activated, which removes the inhibitory subunit from phosphodiesterase like this. So phosphodiesterase activated. This breaks down the CGMP to GMP. So when second messenger is no more, obviously the cation specific channels will not be there. There won't be any influx of ions. So this results in hyperpolarization of the membrane. That means whatever the ions are there, they are going out. Nothing is coming in. So this results in hyperpolarization and the signal passes to the brain. And adding to that, whatever the calcium ions we are present inside, they are also released through the sodium calcium exchanger, thereby reducing the cytosolic calcium level. So this happens during the light phase. Now, when we enter into the dark phase, we already have low calcium level. So this low calcium level will activate the GC and at the same time inhibits the phosphodiesterase because we don't want our second messengers to be damaged or degraded. So PDE is inhibited at one time and guanylate cyclase is activated on the other hand. So this activated guanylate cyclase will be activating the CGMP, reopening of ion channels, thereby returning the membrane potential to the pre-stimulus stage. Okay. Now comes the termination. So this happens in the light phase, this happens in the dark phase. The termination, we have got rhodopsin kinase, which will be phosphorylating the rhodopsin protein. To enhance this phosphorylation, you have got recoverin protein present. Now, this phosphorylated rhodopsin is attracted by another protein known as arrestin. Okay? But after some time, what happens? Arrestin will be dissociating. Rhodopsin undergoes dephosphorylation, and you can see all the transretinal molecules are going out, whereas all the 11 cis retinal molecules are coming in. Now, the rhodopsin is again ready to start up another set of vision reaction. Although when we look into these steps, you can imagine these many steps are there, but everything happens in a fraction of seconds. Okay. Next. Now, this is a kind of match the following. See if you can identify the correct match or not. So you have got ligands on one side, effector proteins on another side. So this is also related to GPCR only. So like this, you can also expect match the following kind of questions in the exam as well. So you have got serotonin, acetylcholine, immunoglobulin E, antigen complex, light. What are their effector proteins? Phospholipase C, phosphodiesterase, adenylyl cyclase, potassium channel, or adenylyl cyclase. I think we have discussed a few of the effectors in some previous questions. So, as previously, Mahesh and Sumita were given correct answers, and I hope so, beautiful colors, you were able to rectify why the option 1 was incorrect, right? So, it's like a learning time. Whenever you are giving a wrong answers, never get disappointed with yourself. It is just a learning period where you are learning something new and ensuring yourself that you will not repeat the same mistake again. That's it. So take it in that note, whoever gives the incorrect answers. Yeah, what is the correct answer over here? Able to identify? Okay, I got one answer that is from Mahesh. Anyone else? Anyone else has any other answer? So that we can proceed with cellular communication part. We are left with 15 more minutes. Sumita, beautiful colors. Any answers from your side? And this is a recent question, recently asked question in your exam. Maybe last time or last to last time. Many a times the match the following kind of questions has been asked in the exam. They will provide you with ligands, second messengers, but in this case they are ligands and effectors. Sometimes they will provide you with the signal transduction pathways. Okay, so the correct answer over here is absolutely correct. It is 3. So we have got serotonin, which serves as a ligand for GS pathway, that is adenylyl cyclase pathway, right? Then we have got acetylcholine, which serves as a ligand for potassium channel or adenylyl cyclase. Now, this is the pathway for GI, right? In GI only, the hyperpolarization takes place. The beta subunit will be responsible for opening the potassium channels. Adenylyl cyclase will be inhibited by the acetylcholine bonding. And in this case, we have got muscarinic receptor instead of nicotinic receptor, right? Then we have got light. Just now I told you that it is related to phosphodiesterase and immunoglobulin antigen complex. So maybe there are chance you may not be able to recognize this. So they are carrying out signaling through the GQ pathway. So all the 4 GPCRs have come over here. Okay. See, allergen binding. The allergen binds to the immunoglobulin E antibodies on the surface of either mast cell or the basophils. After that, this binding leads to the cross-linking of the immunoglobulin on the cell membrane. So upon cross-linking, they will be activating a signaling cascade related to phospholipase C. So when I say phospholipase C, it is related to GQ pathway. So the phospholipase C will be targeting the phospholipids, giving rise to 2 second messengers, IP3 and DAG. As usual, the IP3 will be responsible for releasing the intracellular calcium ions, and DAG will be responsible for activating protein kinases in order to carry out the process. And last stage will be the degranulation stage, that is the increase in the calcium as well as the activation of PKC can lead to degranulation of both mast or basophils, which in turn results in the release of allergic compounds like histamine and other inflammatory mediators, which contributes to allergic responses. Okay, so that is how we can connect it with immunoglobulin E to the PLC. So this is again just a reminder for the Lakshya batch, which started for CSIR NET 2025 batch and GATE 2026 batch. So we have unlimited classes. We have 24/7 chat support system, and a very good fact is molecular biology techniques hands-on. The internship program is also free. Whoever has enrolled into this Lakshya batch. Conceptica series will be there, test series will be there, that will help you to boost your confidence, that will help you to practice more and more of questions so that you will be confident on yourself. So you want to know more about it, you can always call us in any of this toll free number or visit our stores.biotechnica.org. Okay. Next, we have a question from cellular communication. Again, a match the following. Identify which one is the correct combination. So you can take it as option 1 here, option 2, option 3, and this is option 4. Ignore the option IDs. You can give the option 1, 2, 3, and 4. Tight junction, gap junction, plasmodesmata, desmosomes. Very easy and direct question. Let me see how many of you can give this correct answer. Yes, beautiful colors and Mahesh, you were correct for the previous response. So what is the correct combination over here? So column X indicates the junctions and column Y indicates their functions. Tight junction, gap junction, plasmodesmata, and desmosomes. So what is the correct answer over here? I should expect more answers for this question because this is an easy question. And whenever it comes to cellular communication, always expect easy questions. Very rarely you get some experimental question like that. Clarity in the sense you are having no idea about these junctions. Okay, beautiful colors has given the answer. So tight junctions, gap junctions, plasmodesmata, and desmosomes. Connecting the intermediate filaments in one to those in the next seals gap between the epithelial cells, allows the passage of small water molecules from cell to cell. And the last one is small molecules can be passed but not macromolecules, with some exceptions in plants. Options. Okay. This one. 1, 2, 4, 3, like that. Okay, no problem. Let me give you the answer. Okay, Sumita and Beautiful Colors gave the answer. No problem. So the correct answer, what you said is correct. Both of you. So Mahesh, here we go. Tight junctions means what? It basically serves as a barrier. So it seals the gap between the epithelial cells. Gap junctions and plasmodesmata, somewhere it carries out the same function only, but a minor difference. What is that minor difference? Gap junction serves as a channel that allow molecules to pass through it, but it allows macromolecules also. But plasmodesmata doesn't allow macromolecules, with some exceptions in plants. Okay, and the last one is the desmosomes, which is forming a junction that connects the intermediate filaments in one cell to that of the next. Okay, so maybe there can be a confusion between gap junction and plasmodesmata that can make you confused, but be clear with that. Okay, see, I have a table for you. So we have got two types of interactions, cell to cell and cell to matrix. So majority of the interactions are cell to cell interactions only. So adherence junction, the cell adhesion molecule is cadherin, the connecting filament will be actin. These are the adapters, the corresponding function. Desmosomes, desmosomal cadherins will be there, intermediate filaments will be their cytoskeletal attachment. These are the adapters. Hemidesmosomes and focal adhesion, these are cell to matrix interaction. Okay, so in both the cases you will find integrins will be serving as their receptor or the cell adhesion molecule. And hemidesmosomes and desmosomes, they have intermediate filaments only. Similarly, adhesion junction and focal adhesion, actin filaments. Of course, the adapter proteins will be varying and their functions will be varying. Again, tight junctions, cell to cell, occludin, claudin, and jams. Actin filaments will serve as the cytoskeletal attachment, and these are the adapter proteins. Gap junctions, connexin, innexins, and pannexins will be the adhesion molecules. And plasmodesmata, we don't have any specific receptors, but they do have actin filaments and net1A as their adapter. So they are basically communication molecule transport between the cells. Here it involves small molecular transport between the cells. Okay. So this table, if you remember, you can easily fetch a good mark from this 4C. Okay. Now let me give you an experimental question. Cadherins mediate calcium-dependent cell-cell interaction and plays an important role in embryonic development by changing the adhesive properties of cells. Aggregation of nerve cells to form an epithelium is correlated with the appearance of N-cadherins on the cell surface and vice versa. N-CAM, that is neural cell adhesion molecules, belongs to immunoglobulin superfamily of N-cadherin, and N-CAM, immunoglobulin superfamily, and they are involved in fine tuning of adhesive interactions. So, in order to see the effect of mutations, that is, what is the effect of mutation on N-cadherin and N-CAM, two sets of mice were generated. Set A will have the mice with mutation in N-cadherin, and set B will have the mutation with N-CAM. You have to identify the output of this mutation. Very simple. If you know what is the function of cadherin and immunoglobulin superfamily, that is N-CAM, then you will be able to get into the answer. The consequence of mutation will be both the mice will die in the early development. A will die, but B will develop with some mild abnormalities in the nervous system. Or A will show abnormalities and B will die, or both will develop normally and they will compensate for the mutations. What should be the correct answer here? What should be the correct answer over here? Just as I told you, you have to be clear with the cadherin and N-CAM function. With that, you will be able to click into the answer. Okay, Sumita. What about others? And I hope so, Mahesh, you got the clarity of the previous question. Okay, Sarita. Okay, beautiful colors. Anyone else would like to give the answer? Anyone else would like to give the answer? Yes. So the correct answer over here is statement 2. Now, why statement 2 is incorrect? As I was repeatedly saying you have to remember what is the function of cadherin and N-CAM. Cadherins are something that is responsible for mediating cellular interaction at the time of embryonic development, right? See, N-cadherins are critical for early development, especially during the processes like adhesion, tissue morphogenesis, and organogenesis. So that's the reason mutation in the N-cadherin will severely disrupt the cellular communication, specifically the early embryo lethality, as the proper formation of tissues and organs will not occur. On the other hand, N-CAM is a member of immunoglobulin superfamily. So they are involved in the development of nervous system. So embryo will be grown and there will not be any problem as such. But yes, since it is targeted to nervous system, so obviously the mice lacking the N-CAM will have mild abnormalities, specifically in neuronal patterning, connectivity. They will survive, but they will have some defects in the neuronal system. Okay. And I just wanted to remind you that we have got 4 different types of cell adhesion molecules over here: selectin, integrins, immunoglobulin superfamily, and cadherins. So selectins will be recognizing carbohydrates as their ligand. They will not form any stable cell junction. Integrins, on the other hand, will be recognizing any of the ECM proteins as their ligand. So in that case, they form a stable junction known as focal adhesion and hemidesmosomes. If they recognize any immunoglobulin superfamily of proteins, they will not form any stable junctions. Immunoglobulin superfamily of proteins, if they recognize integrins, they will not form any stable junctions. If they recognize the same molecule, that is immunoglobulin superfamily itself, they will not give rise to any junctions. And lastly, if the cadherins interact with their cadherins, that is homophilic interaction, they will be producing a stable cellular junctions, that is adherents and desmosomes. And remember, all the cell adhesion molecules are divalent cation dependent, that is requiring calcium, magnesium or manganese ions for their activation. Okay. So go through this and keep on practicing more and more questions from these topics, specifically the cell signaling part, because from there you always get 2 to 3 questions in every exam. One question in the form of match the following majority of times is expected from the cellular communication part as well. So keep on practicing it and keep on revising the topics till the exam so that you'll get better benefit. And keep on attending this Brahmastra series. As I keep on saying you, this is going to help you out to revise and how to approach a question. Okay. Thank you so much, everyone. See you again in another session with another topic. Prepare yourself and come to the session. Thank you so much, everyone.